This is Richard Radisky from the Annis Water Resources Institute, and I'm going to be talking about harmful algal blooms. Most of the harmful algal blooms in fresh water are from cyanobacteria, and common ones are microcystis, which kind of looks in the center there, kind of looks like a gelatinous bag of cells. There's gliotrichia, which blooms in White Lake, which looks like a furball pet kind of. And then Delicospermum on the far left looks like a chain of beads. Those are the most common ones. And as far as the group of cyanobacteria, there's lots of species and lots of genera, but again, we're only concerned with a small number of the ones that can form toxins and then that are native to the Great Lakes Basin or can bloom in the Great Lakes Basin. They're symbiotic. 8% of lichens contain cyanobacteria. They can fix nitrogen, so if nitrogen is ever in short supply, they can get that from the atmosphere. They're very primitive. They're one of the earliest photosynthetic organisms, but they're very complex in their biochemistry. So they have to do what sophisticated cells like in our body do without organelles. They do it with all biochemical reactions. And they produce toxins, hepatotoxins, which are liver toxins, and neurotoxins affect the nervous system. So that's why we're concerned about them from a public health standpoint. They can adjust their position in the water column by gas vacuoles. Most algae cannot do that. So cyanobacteria can go to the bottom of the lake and get nutrients and then float to the surface. So they can change their position to maximize either sunlight or nutrients. They overwinter in the sediment-water interface. So most algae will not do that, whereas cyanobacteria can overwinter like that. And what's interesting about them is that they can consume more phosphorus than what they need. So we call that luxury consumption, and that means that they can store phosphorus. So gliotrichia, the one that blooms in White Lake, can store a full year of phosphorus or a full growing season of phosphorus that it gets off the bottom of the lake. They also have specialized structures to concentrate CO2, and you can see these granules. So a lot of times when the pH gets really high from photosynthesis, remember photosynthesis takes in carbon dioxide, and carbon dioxide acidifies like soda pop, and the cyanobacteria will take CO2 from the atmosphere and store that so they can do photosynthesis. And then finally, they can live anywhere in the water column and survive at the surface. Most algae cannot take the UV radiation that's right at the surface. They have to live beneath the surface, let the water quench the UV radiation. So they're extremely adaptable, and they've been around a long time. They're some of the oldest photosynthetic cells that I mentioned before. So they're certainly a force to reckon with. As far as blooms, blooms occur when the water temperature is greater than 20 degrees. So when we start getting warmer water temperatures, that's what will trigger a bloom. And it also requires a stable water column. Cyanobacteria do not like turbulent water. They don't like waves. They don't like flowing water. So we need to have a stable, quiescent water column. They like reduced mixing. We know that lakes will stratify due to temperature, and they like that stratification because that reduces the mixing of the lake. They also like reduced flow, so during a drought condition or summer when there's less rain, reservoirs, impoundments, drowned river mouth lakes like Muskegon and White Lake, the discharge is less, so the cyanobacteria like that. There's selective grazing. Zooplankton and zebra mussels will spit out cyanobacteria. There's something about the coating on them or the gelatinous material around them, but zebra mussels are, I guess, smart and they spit them out. And finally, like I mentioned, cyanobacteria will float to the surface, shade out other algae, use CO2 from the atmosphere, and phosphorus they store. And they also have specialized pigments that allow them to utilize the photosynthetic or the light radiation from the sun in a much better manner than normal plants. And this is a nice graphic that talks about the influence of climate change and all the different feedback loops. You can see, the sun, and you can see the cyanobacteria floating to the surface. You can see less mixing. You can see groundwater input of nutrients. And it's just a large feedback loop where the warmer it gets, the more cyanobacteria blooms we will have. Now, there is a counter to that, and it's not totally doom and gloom, is that more frequent storms that we get or more frequent precipitation events will mix the water, and that will break up cyanobacteria blooms. So we are anticipating more blooms, but they may be broken up by episodic rain events. Why do we have nearshore HABs? I kind of like this little exercise to look at them. If you look at the initial distribution, the cells are kind of distributed with light penetration, and you can have like 100 cells and 2 micrograms of toxin, and then all of a sudden they float to the surface. So there's a 1,000-fold or 100 to 1000, 1,000-fold to 100-fold concentration of the cells just by floating to the surface, and that concentration causes more toxin. So there's more cells congregated together, and of course there's more toxin. And finally, the wind will force that entire bloom to one side of the lake. So cyanobacteria don't like storm events, real choppy water, but just a gentle breeze will cause them to be forced to the other side of the lake, and that's where you get the really high levels of toxin. And I mentioned the different kinds of toxins. They have hepatotoxins that cause liver damage. That's microcystins. That's the most common one that we find in the lakes in West Michigan. There's also neurotoxins. Anatoxin A is the common one. We've never really detected that in the lakes around here. It's very unstable. It's been known to occur in ponds, and it's called fast death factor, where a dog will go in the water and get exposed to that material and then have a seizure from the neurotoxin. Again, we don't see much of that here. And then finally, endotoxins or dermatotoxins. These are toxins that cause dermatitis, and the Lyngbia toxin is an example of that. That's in Lake Erie, but we also know that microcystin has a dermal toxin, gliotrichia has a dermal toxin. So, the dermal irritation affects some people more than others. And the roots of exposure are ingestion of the water and inhalation and skin contact. So if you jet ski through the water and there's a bloom in the lake, you will pick that up in the vapor, in the mist, and that will be transferred across your lung tissue and into your body. Just to give you an idea how toxic microcystin is, you can see cyanide down on the scale here, 0.002 milligrams per kilogram per day. That's the reference dose. Microcystin is 0.00003 milligrams per kilogram per day. So it's much more toxic than cyanide. Obviously, it's made in small quantities in the cells, but I just want you to remember that these toxins are significant, and that's why people are concerned about red tides in marine environments. When you have lots of algae, you can get toxin levels that might cause problems. In terms of your activities that can cause cyanotoxin exposure, obviously I mentioned that swimming, water skiing, and jet skiing are the most that cause the greatest exposure. Things like motorboating, windsurfing, wading are kind of moderate. Other exposures like hunting, biking, paddle boarding, sailing, rowing, kayaking, the exposure drops off quite a bit. So just remember that when you're in the water and you're ingesting, you always ingest some water when you're swimming, of course. And then when you're water skiing, you can get aerosols that are inhalation toxins. And one thing to remember is that cyanobacteria blooms are typically patchy. You'll get a bloom in a small pond that'll cover the entire lake, but I've got a slide of Lake Erie, and you can see the eastern basin around the Maumee River and where St. Clair comes in, Detroit River comes in. That's where most of the blooms occur, and you can see that in the middle of the lake you don't see cyanobacteria blooms. Here's a shot of Muskegon Lake, and you can see that the cyanobacteria are congregated near the inputs of nutrients. So they're in the streams, the subwatersheds, Ruddiman Creek, South Branch, North Branch of the Muskegon River. And they're not in the area that has the turbulence from the mixing of the rivers, but when the turbulence starts to go away, that's where you see the blooms occurring. So you don't think of the cyanobacteria as affecting the whole lake, and there's typically always a patchiness or concentrations at a particular spot. The way we assess cyanobacteria is we can screen for them. There's a very strong visual signature when we look at that. We can use remote sensing, test strips, then we collect samples and we test them for toxins. And if we find toxins, we can actually look at the DNA and look at the other different characteristics of the toxins. So there's pretty much a procedure for evaluating HABs and doing tests. We can see HABs with remote sensing. I talked about Lake Erie. We can look for the visible color, or we can look a spectral analysis for the pigments, the specialized pigments that cyanobacteria have. But remote sensing is only good for large bodies of water. And if we applied remote sensing to Muskegon Lake, we'd only have like 1 pixel, and that's not enough definition to see a bloom in Muskegon Lake. We could obviously see something, but it wouldn't give us the detail like where the bloom is in a large body of water. The visual assessment, though, is really strong, and it's a floating green surface scum. And what we have to remember when we see that scum, and I've just got some pictures, here's the scum in the lake, and here's the scum washing on the sand or washing on the beach. Many species of cyanobacteria don't produce toxins, and if it has the toxin producing gene, that gene may not be turned on. So it's a little complicated. However, if you see that scum, that's a warning signal, and you've got to start taking action and doing more testing. We confirm the identification with the microscope. We can easily put it underneath a low to moderate magnification on a microscope and identify it. And that will tell us if it's microcystis, which produces microcystin. It won't tell us if it's a toxin-producing species, but it will give us a first cut that there could be a problem. And then we, of course, need to do laboratory testing. And like I mentioned, there's field tests. There's little test strips that you can purchase and just put in the water. And that's how the eagle typically responds to cyanobacteria blooms. They will get a report from beach monitoring or from the public. They see the scum forming on the surface or on the beach. They will send somebody, or the public health department. Public Health Muskegon County has been trained in using the test strips, so they will respond, take a measurement right at the site, and then if the measurement is positive, then they will send the sample for analysis. There are imposters, and it does take a little bit of a trained eye. Cladophora, forms surface scums, but it's really stringy. Pollen, there's some lakes in Muskegon County that we monitor, like Fox Lake, that for whatever reason has a lot of pollen, and that's more yellowish. So it's a quick microscope, put it under the microscope, and you can tell pollen grain very easily from cyanobacteria. And then duckweed. Duckweed forms in wetlands, but when you look at it, it's the smallest angiosperm plant, so you can tell right away. So there are imposters, but microscopic identification will reveal if it's a cyanobacteria or not. It takes some training to identify cyanobacteria. Eagle offers a class for water plant operators, and we're trying to get that offered for public health professionals. But again, we're dealing with just a few species, and you can recognize, the one that forms the chains, the Delicospermum. You can recognize the colonies, the Microcystis. And then there's one called Oscillatoria, which are the long rods. That's not a toxin-producing organism. So if you have a little bit of taxonomy background, and generally one day going over taxonomy, you can do these kinds of analyses. The other nice thing you can do is you can take a picture of that right through the ocular. This is an ocular photograph with a cell phone, and you can send that to an expert. I do a lot of ocular photo identifications in the summer. People, take a picture like that and send it to me. And then there's a company called Bloom Optics that has a portable microscope, and it interfaces with your iPhone. And they actually are using a artificial intelligence software program that will identify the cyanobacteria and count it. So you put it under the microscope, take a picture of it, send it up to the cloud, and within a minute you get an identification back and a number back. We tried that in parallel with our class last year, or excuse me, this year, and got really good results. I mean, the water plant operators were able to use the Bloom Optics microscope, and we got good results between identifying it the old-fashioned way and using the Bloom Optics. So it's a little pricey. It's $1,000 for this technology, but if you don't have access to a person that knows cyanobacteria, and you want to do this in the field, this is field portable. This is a nice option to consider. I wanted to show you some data from Muskegon Lake, and in 2021 we had heavy spring rains and warm fall temperatures, and we started to see cyanobacteria colonies at Pier Marquette Beach and North Muskegon beaches. So they were coming out, the bloom was occurring in Muskegon Lake, and it was washing up it was coming out the channel and washing up on the beaches right near the Muskegon River channel. Microcystin was detected in the Muskegon drinking water intake. That was the first time. It was 100 times below the drinking water limit, so it wasn't serious. And it was only in the raw water. The chlorination, the treatment process removed whatever microcystin was there. But that was kind of a wake-up call, because it was the first time, and they test Muskegon intake water a couple times every year. That was the first time it was detected. So harmful algal blooms in Muskegon Lake can be advected or moved out into Lake Michigan, and they can't really survive out there for any extended period of time because the waves are too strong. But if you have quiescent periods of time, calm periods of time, it's just something to watch. And we were trying to work with the Muskegon drinking water plant to put a chlorophyll sensor, a phycocyanin sensor in their water system. We couldn't get the money for it, but anyway, there are ways to screen the water and make sure that you don't have cyanobacteria present. But we did have very high levels of microcystin in the nearshore areas of Muskegon Lake, and there was actually a shift between a non-toxin producing organism, which is called Microcystis wessenbergii, to a toxin-producing species, Microcystum aeruginosa. So for some reason, the population shifted in Muskegon Lake at the same time that we had these heavy spring rains and warm fall temperatures. And we were able to get NOAA to come and do a hyperspectral flyover, and they actually flew over the lake, and you can see all the pictures that they stitched together. And you can see where the cyanobacteria blooms are. And here's a, excuse me, here's a hyperspectral picture. This is obviously enhanced, and we don't know the concentrations here, but you can just see how patchy the cyanobacteria blooms are. And the water temperature was warm. It was almost 70 degrees well into October. and we had spikes of the cyanobacteria chlorophyll. This is a shot in one of the marinas. You can see the cyanobacteria bloom accumulating. And as far as concentrations, microcystin LR is the one that the variant, toxin variant we're concerned about. Drinking recreational water is 8 micrograms per liter. Drinking water is 0.3 or 1.6 depending on the age. But we were into the thousands at Fisherman's Landing, which is a boat launch. Water Sports Park wasn't quite that high, but you can just see we had some very high levels of microcystin in these blooms, and they were congregating in that near shore environment because the wind blew them there, and they were just kind of festering in that environment. So summary, HABs events will continue to increase due to climate change. HABs can contain toxins that cause dermatitis, liver failure, gastroenteritis, and neurotoxicity. They're patchy though. Remember that the whole lake is generally not contaminated, and the highest levels accumulate on the shoreline. And they've got a very strong visual signature, so the best advice is to avoid contact with the water. Sometimes kids like to play in that green scum, and if your parents, tell them no. But when you see that scum, the public health department or Eagle needs to be notified to investigate. So the people that helped contribute to this presentation were Eagle, NOAA, the Michigan Department of Health and Human Services. They're the ones that have the testing lab for the cyanotoxins. Public Health Muskegon, they're the ones that we do the beach monitoring for, and like I said, they were trained on using the test strips so they can respond. And then finally, the Water Resources Institute. So thank you.